Samirini Kalapala, pursuing my BTech Biotechnology at KL University, Vadeswaram. This video is about polymerase chain reaction. So let's get started. Polymerase chain reaction is a technique to make many copies of a specific DNA in vitro. Rather than series of manipulation involving living cells, PCR is carried out in a single test tube simply by mixing DNA with a set of reagents and placing the tube in a thermal cycler, a piece of equipment that enables the mixture to be incubated at series of temperatures that are varied in program manner. The basic steps in PCR are well explained in this video. So watch till the end. In the PCR, the reaction is repeatedly cycled through a series of temperature changes which allow many copies of the target region to be produced. PCR has many research and practical applications. It is routinely used in DNA cloning, medical diagnostics, and forensic analysis. Let's talk about principles of PCR. PCR typically involves a series of 20 to 40 repeated temperature changes called cycles, with each cycle usually comprising 3 discrete temperature shifts. First one is denaturing at 94 to 96 degrees Celsius, then primer annealing at 45 to 60 degrees Celsius. Primer extension is the last one, usually at 72 degrees Celsius. The most of PCR methods typically amplify DNA fragments up to 10 kilo base pairs. However, the same technique can amplify up to 40 kilo base pairs as well. So, move on to the components of the PCR. Setting up a basic PCR requires many ingredients, reagents, and conditions, which are enlisted as follows. The target or template DNA, the primer is DNA polymerase, nucleotides, buffer solution, monovalent and divalent cations, PCR tube, thermal cycler or thermocycler. Let's move on to the procedure. The first step is denaturation, in which we are going to heat the reaction strongly to separate or denature the DNA strands. This provides single-stranded template for the next step, followed by annealing, where we cool the reaction so the primers can bind to their complementary sequences on the single-stranded DNA template. The last step is extension, where we raise the reaction temperature so Taq polymerase extends the primer, synthesizing new strands of DNA. This cycle repeats 25 to 35 times in a typical PCR reaction, which generally takes 2 to 4 hours, depending on the length of the DNA region being copied. The reaction is efficient. The target region can go from just 1 or a few copies to billions. Let's talk about types of PCR. These are the different types of PCR, starting from conventional PCR, multiplex PCR, nested PCR, real-time PCR, also called quantitative PCR, colony PCR, assembly PCR, or polymerase cycling assembly, touchdown PCR or step-down PCR, hot start or cold finish PCR, methylation specific PCR, inverse PCR, and lastly reverse transcription PCR, which now become famous as RT-PCR from corona pandemic. Advantages of PCR. PCR is highly sensitive compared to culture and staining techniques. It is performed in 4 to 8 hours. It is very quick. It is shown to be more cost effective with selective use than culture and staining. Increased ability to detect less common organisms such as virus. Ability to test for antimicrobial resistance are the advantages of the PCR. Like every technique, PCR also has some limitations. So the limitations of PCR are that the DNA polymerase used in PCR reaction is prone to errors, can lead to mutations in the fragment that is generated. The specificity of the generated PCR product may be altered by non-specific binding of the primers to other similar sequences on the template DNA. In order to design primers to generate a PCR product, some prior sequence information is usually necessary. These are the imitation, and that's the end of the video. Thanks for watching, and please leave a comment and like this video. Thank you.